Hi, my name is Marguel, um, and the project that I'm going to present is crispr-ga. crispr-ga is a tool to analyze gene editing results. Um, so basically, this comes from a project that we published a while ago in the lab that I'm working with at the genetics department here at Harvard Medical. So basically, this work tried to provide with a tool to analyze gene editing experiments, and basically, from a general perspective, we provide some kind of representations that help the user to assess what was the efficiency and what was the off target of the gene experiment gene editing result that he performed. So all this tool is implemented in a website. This is the website. Basically, in this website, the user provides with the sequencing data from the cells that has edited. Then the user pastes the sequencing information that he wants to implement in the cells, and then the reference, which is the current sequence of the cell. Then basically he submits the results. He submits the work using the button upload, and after a while he will get a report, and this report basically gives all this information of basically which type of editing he got, like deletions, insertions, and also where this editing is localized. So as you can see, this output is not. It could be way improved. So is what I am trying to do in the final project from the visualization class. So this is the new design of the output that the user would receive. Still, it has 3 panels. So one of the panels is the main visualization of where the gene editing happen. So basically, in the x-axis we have the locus, and in the y-axis the depth of editing. So then we have another panel that basically informs us of the distribution of how big were the different edits. So this in the y-axis we have the size, and in the x-axis how many. And finally, all these are distributions, and here we have the exact reads that generated these results. So basically, each of the lines consists of one read, one sequencing read. The green are the deletions, and the red the insertions. And basically, the exact bases that are deleted are the black lines, and the exact bases that are inserted are the red dots. Then I implemented a brushing function so that we can select certain areas of interest. For example, if the user is interested in seeing what composes the gene editing that he got in this parcel, he can select, he can move around. So, and then basically the 2 other panels are updated in synchrony. Another functionality that I implemented was basically to simplify the plots by either only plotting deletions or insertions. For example, in the case of selecting deletions, then whenever basically we select a region, we only get deletions. And the same we could do for insertions. So for example, then we only get the insertions. Then another thing that I implemented basically is this pie chart. This pie chart provides us with the results, the overall results, the non-homologous adjoining NHEJ, so the percentage, and also the percentage of HR, which eventually is what the user will be very interested. And then in terms of more general functionality, I tried to include pop-up messages to help the user, for example, a message to tell what's in this panel, another message to explain what is in this panel, and the same with in this panel, and the same with the results. So yeah, and I think that was like a summary of how the tool works. Yeah. Thanks for listening.